In this video, let us discuss about production of monoclonal antibodies. Remember, it was a discovery in the 1975 that has really revolutionized in the bioscience research. If we produce antibodies in a laboratory, how do we produce that? We inject an antigen in a particular animal and then we wait for a few days so that antibody is produced. Assume we are injecting an antigen into an animal. So now an antigen will have several antigenic determinants. Assume this is the antigen, and this antigen has one antigenic determinant like this, another antigenic determinant in this manner, another antigenic determinant this way, and a fourth antigenic determinant in this manner. After few days, when we collect the antiserum from this animal, you can get the antiserum having an antibody against, assume, let us number this as 1, this as 2, this as 3 and this as 4. There will be antibody number 1 which is specific against this antigenic determinant. Antibody number 2 specific against this. Similarly, antibody number 3 and antibody number 4. This is what we are going to get. Which means in the antiserum which we got it is a polyclonal antiserum. It has developed antibodies against each of these antigenic determinants. Now the question is, suppose you want to purify only one of these. You want to purify only this antibody against antigenic determinant 1 only. This is going to be a laborious process. Why it is a laborious process? It is like looking for a needle in a haystack. Or from among all these polyclonally multiplied cells from the antibody collection, identify that antibody because it will give a very, very low yield first of all. And there is a possibility of cross reactivity. Several times you have to pass through these antibodies through an absorbing medium and to separate it. And really a laborious process. It is in that context, two scientists thought about, is it possible to develop or develop only one particular clone so that monoclonal antibody can be created. So their thinking went on in the line of, for example, if this is mixed with myeloma cells. Myeloma cells are cancerous plasma cells. So mixed with the myeloma cells. Now what happens with the myeloma cells is, each of these antigenic determinants can fuse with, for example, this is the myeloma part, this is number 1, this is number 2, this is number 2, this is number 3, and this is number 4, and this is the myeloma part. Fusing normal B cells, antibody producing cells with the myeloma cells, which can give a long lifespan. And that will be able to produce, suppose you are able to select only this number 1, then it is capable of continuously producing large quantity of monospecific antibodies. This is the concept that we are going to develop as the production of monoclonal antibodies. Now, this was, as I mentioned, this was a concept that developed in 1975. Two scientists, and their names are, one is George Kohler, and the other one is Caesar Mailston. George Kohler and Caesar Mailston, they carried out several experiments in order to produce monoclonal antibody. What exactly they did is they primed the B cells of an animal. So they took primed B cells. Primed B cells means a B cell which is primed with a very specific antigen. And remember, it will have different epitopes. From that, they are going to select a particular clone and going to amplify or are going to culture and produce monoclonal antibody. So they took primed B cells and mixed with the cancerous cells. So the cancerous cells are nothing but they are also the plasma cells but coming from a cancerous. What is the problem with the B cell? The problem with the B cell is it has short lifespan. They will die after some days. Whereas the cancerous cells, they are immortal. They are immortal. They can live unceasingly. This is the property that is used in the production of monoclonal antibody. What they did is they fused these cells together. Fusing the cells is very simple. What can be done in fusing the cell? You can use a chemical. A chemical like polyethylene glycol can be used, or sometimes you can use an envelope virus known as centivirus can be used. So when you use these substances to fuse a cell, what is likely to happen? You have a B cell, you have the myeloma cell. Possibilities are either some cells will not fuse at all. So this is an unfused B cell. And some myeloma cells also will not fuse. Let us write M. Unfused myeloma cells. They will not fuse. But there are possibilities. B cells and the M cells, the myeloma cells, can fuse in three ways. One possibility, there can be a fusion of one B cell with another B cell. What the polyethylene glycol does is, it will fuse the plasma membrane together and all the cellular content becomes together this way. So therefore there can be fused B cells here and there can be fused a B cell with myeloma cells, and the third possibility is there can be two fused myeloma cells. These are the various possibilities. Our interest is in fused cells. Now in these fused cells, we shall call these fused cells as heterokaryons. So they are called either heterokaryons. Now over a period of time, if the unfused B cells will die, if the unfused M cells, we are not interested in that. We are interested only in these heterokaryons. How do we select in the right cell which has the capacity to produce monoclonal antibody? We know that these BB cells are not important for us because it will not be able to give us, it will not last longer, it has a short lifespan. This MM cell also the same. They are immortal, but it will not be able to produce the desired monoclonal antibody that we want. We are interested only in this cell. Now let us continue understanding this based on these kind of heterokaryons. So we have these heterokaryons, which is if the B and the M, if the plasma membrane is fused together, if the cellular contents are fused together. Milestone and Koehler used a very, very interesting knowledge at that time. That knowledge is the knowledge in nucleic acid metabolism. If you look at the nucleic acid metabolism, nucleic acids can be synthesized in two ways. One is called a de novo pathway. A de novo pathway, it means it is beginning, synthesizing from scratch. So in de novo pathway, what is done is, we have a molecule called PRPP. phosphoribosyl pyrophosphate. Phosphoribosyl pyrophosphate that can be added to a uridylate, a nucleotide. Now you are able to synthesize thymidylate, and thymidylate is required for nucleic acid synthesis. So this you need for nucleotide formation and for the synthesis of DNA. Only if synthesis of DNA happens, cells can multiply and then they can grow. Therefore, this particular step is very important. So this is how thymidylate is produced. Another way to make nucleotide is you have something called a salvage pathway. What happens in the salvage pathway? It is salvage pathway, all the broken pieces are brought together, assembled, and nucleotide is made. So in salvage pathway, hypoxanthine is used. You can use thymidine, hypoxanthine, thymidine. These can be used in order to make a nucleotide. To that, PRPP is added and nucleotide is made that is used for synthesizing DNA. Now, for synthesizing this, you need two enzymes. One enzyme is known as HGPRTase, hypoxanthine guanine phosphoribosyl transferase enzyme. Another enzyme is TK, that is thymidine kinase enzyme. Now remember, these two enzymes must be present. And where are these gene for these enzymes? They are present in the B cells. So therefore, this is HGPRTase positive and TK positive. It should be present. Now, another important aspect is in the de novo pathway. In the de novo pathway, it is active in all the cells. It is active in the B cells as well as it is active in the myeloma cells. But we don't want in the myeloma cells, we want only the immortality of the myeloma cells. We don't want the de novo pathway to be active in the myeloma cells. So therefore, how do we do that? Very simple, use a substance known as aminopterin. Aminopterin is a structural analog of tetrahydrofolate. You know that tetrahydrofolate is a substance which acts as a methyl or a formyl donor in nucleotide synthesis. If you add to uridylate, if you add a methyl group to uridylate, it becomes a thymidylate. So 5-methyl uracil is thymine. So that methyl group is provided by tetrahydrofolate. So it is able to synthesize nucleotides and synthesize DNA. We are stopping it. How do we stop it? By adding aminopterin, which is a structural analog of tetrahydrofolate. So no de novo pathway will occur, only salvage pathway will be promoted. Salvage pathway means in the B cells, salvage pathway is promoted, so B cells can keep on multiplying. Now, therefore, in order to ensure there is no cross-contamination, if the myeloma cells are now, it is immunoglobulin negative. They should not produce immunoglobulins. If it produces immunoglobulins, there is a possibility that is you will get hybrid immunoglobulins. So initially when Kohler and Milston carried out the production of monoclonal antibody, they produced a hybrid monoclonal antibodies. Later on, this gene was knocked down and that's why they could develop pure monoclonal antibodies. So it has to be Ig minus and it has to be HGPRTase minus. Myeloma cells should not carry out in the de novo pathway. So myeloma cells, we are taking only one property, that is the capacity of immortality. From B cells, we are taking the property of cell division, capacity to produce antibodies. And you see how this particular logic is used in order to select the fused BM cells. In order to do this selection, there is a media prepared, and that media will have aminopterin, it will have hypoxanthine, and it will have thymidine as a substrate. That is known as HAT medium. Hypoxanthine, aminopterin, and thymidine medium. It is called HAT medium or the HAT selection. Let us rub this one and let us see how we proceed with that selection. So far what we have done is we have primed the cells, fused it with the myeloma cells, we produced the heterokaryons, and from the heterokaryons we want only those heterokaryons which will produce antibodies of our interest. Now what is going to happen is you have a HAT selection. HAT selection is carried out. In the HAT selection, that is over a period of 7 to about 10 days, all the B cells, they will die because they don't have immortality. In the myeloma cells also will die because they are HGPRTase minus. They cannot survive. They will die. Now, there is a lot of rearrangement, cell division happens. Please understand that these are not the only two nucleus that will be present in the heterokaryon. There will be 7-8 nucleuses present in a heterokaryon. So that many copies of chromosomes will be there, which means in the process of cell division, many of the heterokaryons will die. Many of them will die, but some will survive. And some will stabilize after a number of cell divisions. It will stabilize into one nucleus. And this one nucleus will have a mixture of chromosomes from B cells as well as from the myeloma cells. And that is a stable hybrid cell. And this is known as hybridoma. So we have produced hybridoma cells. Now it's a most important aspect. Hybridomas are created. Now from this hybridoma, we come to the next stage. In the next stage is we need to select which clone is capable of producing the antibody is of our interest. Let us see how do we do that. That is the second stage. After the HAT selection, we have a hybridoma. So it is HGPRTase positive. It is capable of producing antibodies and they are immortal. This is what the HAT selection has done, which means B cells, they have taken these properties from the B cells. Only the B cells and in the myeloma cells with the immortality are capable of surviving or producing antibodies. Now comes the second stage. In the second stage is screening. Screening, they will be grown on plates and we should be able to screen each clone. Each clone of cells is screened for production of antibodies. Now, initially it will produce very small quantity of antibodies, producing in nanograms of antibody it will be produced. Now, the screening is carried out by using ELISA or RIA. Both are very, very effective method of screening methodology in order to identify which clone, which fused cells or which hybrid cells are capable of producing antibody. It is a laborious process in this particular screening methodology. Once the screening is done, we reach the last stage of antibody production, that is its propagation. Propagation, initially when Kohler and Maidstone done production of monoclonal antibodies, they were able to produce only in micrograms of antibodies, but the technique was proved. Now, propagations can be, it's normally in the initial period, it is done in two ways. One way to do propagation is, it is a cell culture. Cell culturing is done, you know the cell culture vessels, in that propagation is carried out. Another way to propagate is in a immunocompatible animal. Take an immunocompatible animal and inject into the peritoneal cavity. In the peritoneal cavity, when the fused cells are injected, the antibodies are secreted into the ascites fluids. So antibodies are secreted, and these monoclonal antibodies can be continuously segregated or separated, and this is capable of giving in milligrams of monoclonal antibodies. These were the initial stages of its separation, but of late we know that monoclonal antibody cell culture is possible even in large fermenters as big as 10,000 liter capacity chambers. In fermenters, monoclonal antibodies are produced. What we have seen very quickly is what is the principle involved in its production, how a hybridoma is produced, the role of the hat medium in producing that, and then how it is screened and then it is propagated.